going is we've got a situation that we've got some stuff in the stomach. Now, in the stomach, we know we've got acid, hydrochloric acid, and we have an enzyme called pepsin. And we can do an experiment to have a look at what happens when we have different pHs. So let's have some test tubes. So let's zoom in a little bit. There we go. Right, I've got stomach, I've got test tube. Now let's have a look at what we can do with different conditions here. Well, I think we need to set it to about 37 degrees centigrade. And at this temperature, the enzyme will work probably at its most efficient. And what we can do is we can take a piece of meat and we can put that into each of these test tubes. Into the first test tube, we can put just HCl. And we'll see the effect of HCl on the reaction. We'll put the other one into pepsin, just with pepsin. So some water and some pepsin. Can't spell. And we'll see what happens with this pepsin. But the pH of this will be about 7. And into the final one, we can take some pepsin plus some hydrochloric acid, and that will set our pH down probably around about pH 2. And we leave this for about 2 to 3 hours, the time it can take for you to do your digestion. And what are we going to find with this experiment? Well, what we should find is that we've got 3 sets. We've got HCl, we've got pepsin, and we've got pepsin plus HCl. And what we should find, if we leave these going for a little while, is more or less nothing happens to meat in acid. It does break down, yes it does, very slowly, so we might see some slight breakdown, nothing, or maybe slight. With pepsin we'll see slight breaking down. Pepsin will work on the meat and it will try and break it down, but it's at the wrong pH. The enzyme's not going to be working very efficiently. Only when we've got pepsin working at the right pH will we see some breakdown. And it's because this enzyme works at this particular pH. So we can ask a question, why this pH? Why do we want pepsin working at this pH? So the stomach's known for doing 2 things. One, it produces acid, hydrochloric acid, and one of the effects of hydrochloric acid is this is going to destroy any sort of bacteria that's going to get into your system. And in fact, a lot of your defense system involves taking anything nasty and shoving it into your stomach where it's going to be destroyed in acid. And the other job of it is to produce this enzyme, pepsin. It's a clever enzyme, in fact. It's made as an enzyme called pepsinogen. And pepsinogen doesn't do anything, which is good because pepsin dissolves protein. and this is now inside the cell of your body. When it's released into the stomach, it comes in contact with the acid, and the acid turns pepsinogen into pepsin, where it can start digesting the food. So it's quite a clever design system. So this pepsinogen is relatively safe and not causing your body any problems. Put it in the stomach where the acid is, and then it's turned into pepsin, where it will start digesting any protein. And I said, we can't really do this experiment. As it leaves the stomach, we end up with a picture, I suppose, here. Let's just have my stomach. I'm not very good at art, as you are only too aware. It comes out of the stomach and it goes into a bit of the small intestine, and this is called the duodenum. And into this duodenum we have an organ here, the liver, and the liver has a little gallbladder. And the gallbladder here, it supplies bile. Now, what's the job of the bile? Well, the bile simply takes the pH of 2 and we increase the pH to round about pH 7. I think I am going to have to just zoom out a little bit to make life easier for myself. Right, there we are, you can see. Bile will take everything to pH 7, where all the other enzymes can work quite happily in the body. All the other enzymes seem to work around about pH 7. It's just pepsin with its job of breaking down the proteins and perhaps getting rid of the bacteria that needs pH around about 2. The other point of having the bile is that it acts as an emulsifier, and it emulsifies the fats. What do we mean by emulsifying fats? Well, something that you're probably aware of, that if we take some oil and we take some water, then they don't mix very nicely. Let's have a look and see what we can do with this. So let's take a look. I've got my heart ready to go in a minute. That's all set up. But what I want to do to start with is just to take here a little conical flask, and what I'm going to do in here is I'm going to put some water. There we are, a little bit of water in there. Hopefully you can see that. And I'm going to put some oil in here. Now, if I put the oil in here, we can see that the oil floats on the water. Let's put a bung on here, and what we're going to do is we're going to shake it up well, and we get this white colour sort of emulsion. But the sad news is, if I leave this, then what's happening, and I'll see if I can zoom in here just so you can see. We're starting to form two layers. We're getting the water layer at the bottom and the oil layer at the top. Now, if my enzyme is water based, it's not going to get to this. So what we need to do is to put in some bile, and I just happen to have some bile substitute here. And here's my bile substitute. It's washing up liquid, and just before I put this in, we can see that we've actually got quite a yellow layer of oil with the water, and although I shook it up, it didn't stay like that. So let's just put a squirt of washing up liquid in there. Let's put the lid on and we'll give it a good shake. And if I leave this, it's going to stay more or less like this. The washing up liquid has enabled the water and oil to mix, and if I take a little sample of this out, then we can see that when we try and get this out, it's just lots of little droplets of oil surrounded by water, and this means the enzyme can get at this. So there is a very, very simple experiment just showing how you mix oil and water. That I've got another organ here, the pancreas, and the pancreas simply enables us to put in enzymes into the system. And the pancreas is going to produce basically three different enzymes. It's going to produce a carbohydrase. So let's just push this up a little bit. So it's going to make a carbohydrase. The carbohydrase is going to break down the carbohydrate. And it's going to break that down into sugars. We've got another enzyme in here called a lipase, and that's going to break our fats down into fatty acids plus glycerol. And finally, we've got a protease, and that's going to break down any remaining proteins or bits of proteins called polypeptides into the amino acids. And these are our building blocks that can be used to modify the system, to build new bits of you. Basically, the idea then for getting digestion to work properly, we need a suitable temperature, we need to have a suitable pH, and we need to have a large a look at the heart. I've got my model heart here, and it might be fairly hard to have a look at it, and it's hard to see some of it because we don't have an easy way of trying to move bits and pieces around. So what I have got is a small amount of dye which I'm going to try and put into the system. So let's have a look at this system first, and then I'll try and put a little bit of this dye in and we'll see what happens. So let's start with this here. I've got a 4-chambered heart. I've got here, this side, and if I try and look at the way a heart works, this is on my right side if I stand behind it, so it's going to be the right atrium, and blood comes into the here. It then goes into the right ventricle where it's blasted out here and goes either this way or that way to my lungs. My lung seems to have fallen down here, so I've got my couple of lungs. They don't look much like lungs really, and the blood having entered these lungs then returns to the left side of the heart, the left atrium, where it goes into the left ventricle, where it is then pumped out round the whole body, either through the lower part of the body itself, the legs, or it's pumped out to the arms and the head, and then all of that returns to this left atrium, right atrium. Right, let's try it in and see what happens. We might be able to see this, but I'm going to add some dye to see if I can make it better. And if I get it going, we might just be able to see this water entering the atrium here, and you might be able to see it filling up. Can you see that? Or might it be better if we put a little bit of dye in there? Yeah, now I tend not to put too much dye in here because it tends to make one awful mess, but we'll be kind and we'll do it for you. So we're going to put this little bit of dye in here. This is Congo red, and it's particularly messy. So I've got my little bit of dye, and we're going to try and squirt some of this into the system. So I'm just going to take a little bit of this dye, and what I'm going to do is I'm going to try and put it in here. And I'm going to fail, so I'm going to put it into this part of the body, and we can see it just entering the atrium. Right. As I said, this is going to make a lot of mess. Right, we've got the blood in the right atrium. I'm going to pull this back up so hopefully we can see what's going on. And as I now move this blood, it's going to go into the right ventricle. As we pump again, this is going up to the lungs and it returns to this side, the left atrium, and it's now going into the left ventricle, where it is now being pumped around the rest of the body and returning. And we see that after a little while all this blood now looks the same colour as I'm mixing it all up and passing it around the body. But you can see we've got it squirting here into the atrium. We can see it here squirting into this atrium and filling up and emptying. The ventricles are staying more or less full, and we can see that the lungs are relatively full. Got to keep a good heartbeat going, but hopefully you can see this working reasonably well. There we are. I of course now seem to have blood all over my hands. Of course it isn't blood, this is Congo red, which is quite a nasty stuff, and I can leave this lying around, and it's used for false blood. Can you pass me some tissue? And I can mop that up fairly quickly and easily, and then I won't get into quite as much trouble. There we go. Yeah, I think I'm probably still in trouble, but I'll go around with my little red fingers for the rest of the day, because that sort of dye stays on there.